Now, what are the topics that we are supposed to do? One is our DNA fingerprinting.
Human genome.
And Human Genome Project. DNA fingerprinting and Human Genome Project. Okay. So both the topics are at the end of the chapter. Your molecular basis of inheritance. Overall, once you basically when you reach this topic, you're already done with transcription, you're already done with translation, and you know a little bit about genetic code. So a little bit knowledge is there with respect to what the DNA is, what the structure of DNA is. But most importantly, if I want to study these two topics, I also need to know about tools of biotechnology. Tools of biotech. What are the tools? We need to know about restriction enzymes. We need to know a little bit about PCR, how to isolate DNA, and what is autoradiography, and stuff like that. So the general trend of teaching the topic is you teach MBI till regulation of gene expression. After that, you start with biotech 1. Once you're done with biotech 1, then you can come back to DNA fingerprinting and Human Genome Project. So I believe that your guys' biotech 1 is done, right?
Yes, sir.
So biotech 1 is done. That means you know about the tools of biotechnology. You know about restriction enzymes and all those other things, right? Yes or no, bolo bhai.
You have a fair idea.
You have a
Yeah.
fair idea. That is more than enough. For this chapter, it is more than enough. Now, what is DNA fingerprinting or what is DNA profiling? DNA profiling or DNA fingerprinting. One thing that you need to understand is that every organism has a unique DNA profile or a unique DNA fingerprinting. Okay, and this DNA fingerprinting does not relate to this wala fingerprint. Although fingerprints of two human beings are also not seen, you can apply the same logic that similarly the DNA fingerprint is also not seen. What do we do in DNA fingerprinting? We identify those points within the human DNA which are different, and we use that to compare. Generally, the use of DNA fingerprinting is there when you are looking at a crime scene. You want to figure out who has committed the murder. We are going to use that here. We use it to identify, you can say, paternal testing, when you have to figure out who the father is in that case. So yeah, that is where you use DNA fingerprinting. Now, first question that is asked is, who gave this, who discovered it? So what is the answer to that? It was discovered by Alec Jeffrey. Okay. Now, what did he do? What did he use? And what was his concept behind DNA fingerprinting? First thing is, when you study Human Genome Project, you already understand that 99.9% of the DNA is same in all organisms. Of DNA is same in all organisms. What is the different? The different part is just the 0.1%. Okay, 0.1% is different. Now you might be thinking, sir, 0.1% is such a small amount. But if we look at human genome is 3 into 10 to the power 9 base pairs. And out of that, 0.1% of that human genome is, how much beta? What is 0.1%? 0.1 by 100.
3 into 10 power 6.
Into 3 into 10 to the power 9, which comes out to be 3.3 into 10 to the power 6. Clear?
Yeah.
Is it clear? So the whole study is going to be on this particular 3.3 into 10 to the power 6 base pairs. Is that fine?
Yes.
Okay. Now, when we look at a chromosome, okay, what are we looking at? We are looking at a chromosome. And let's look at all the chromosomes that are there. Okay, you have to look at chromosomes. So within the chromosome, there are two types of DNA. Within the chromosome, there are two types of DNA. A is the bulk DNA or the genomic DNA. And B is called the satellite DNA. Question is, what is this satellite DNA? The satellite DNA is a particular part which is near the centromere and telomere. Near the centromere and telomere. Is that okay?
So isn't it after the telomere?
Yeah, that part which is after the telomere, that is also satellite DNA. And some part near the centromere is also the satellite DNA. Both the places you find satellite DNA.
Okay.
When we study cell the unit of life chapter, when we go towards the chromosomal structure, there it is shown. First there is telomere as the secondary construction. After that, whatever is there, that is your satellite DNA. But we need to understand कि जहाँ पे centromere होता है, there also there is satellite DNA. Wherever there is a telomere, there also satellite DNA is there, and satellite DNA is also dispersed throughout the chromosome as well. So it is everywhere. What is satellite DNA, beta? It is basically a type of repetitive DNA. Is that clear?
Yeah.
And it is non-coding in nature. Repetitive DNA, which is non-coding. So it is not making any protein. The whole concept was we have DNA which can make protein and DNA which cannot make protein. So, sir, if Sir Alex Jeffrey or Mr. Alex Jeffrey wanted to study this, he needs to separate out the satellite DNA. How do you do that? It's very simple. Take a test tube. Do the same thing which was done by Messelson and Stahl. The cesium chloride density grade centrifugation. Cesium chloride density grade centrifugation. And when you do that, within the test tube, you will have two sets of DNA. One will be the lighter part and one will be the heavier part. This is your satellite DNA. And this is your bulk DNA. Now, if I draw it on a graph, there will be two peaks in the graph. One will look like this. And one will look like this. This is what represents the bulk DNA, the heavy part. And this is what represents the satellite DNA. So one thing till now, which is clear to everyone is that there are two types of DNA. There is bulk DNA and there is satellite DNA, which can be separated out. Other than that, which something else which is also clear is that satellite DNA is repetitive DNA, which does not exactly code for anything, and it is found near telomere and centromere. Any doubt till here?
What is the graph, like x-axis and y-axis of the graph?
Excellent. I think weight, density, and weight, like that.
Oh, I thought you said weight. Okay, okay.
Now, if you look at the other part, sir, what is repetitive DNA? We are going to study the satellite DNA. DNA fingerprinting is based on satellite DNA. So what is this repetitive DNA? Let's understand that. Let's say we have an individual. Let's call them individual X. Now, when we are studying this individual X, we have their chromosome number 7. And in their chromosome number 7, we notice that there is a particular patch where we see
So wait, can you just repeat what you just said? The individual
I
X
said
thing?
that we are studying a particular individual X, and in that particular individual X, we are studying their chromosome number 7. And in their chromosome number 7, we are seeing that a particular sequence, which is ATC, 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 it is getting repeated. Is that clear?
Yes, sir.
So if I draw it, it will look like this: ATC, ATC getting repeated again and again. Now, what is this ATC? This ATC is one repeating unit. What is it, beta? One repeating unit.
Yeah.
And how many bases are there in this? 3 bases are there. And how many times does it repeat? We can count 1, 2, 3, 4, 5, 6, 7, 8. So how many times the repeat is there?
8.
8. Now, if we are going to individual, let's say Y. We are going to individual Y now. In individual Y, we are studying the same thing, the chromosome number 7 again. Now, in chromosome number 7, what we notice is, let's say this guy has the same repeat, which is of ATC. Although the repeat can also change, but let's say they have the same repeat, which is ATC. But the amount of time ATC is there, now that has changed. Instead of being repeated 8 times, now in this person, it is being repeated only 6 times. So now what do we write? We write the same thing. 1 unit, 3 bases, and number of times it has repeated is equal to 6 times. So what we notice from this overall thing, we are seeing that the satellite DNA or the repetitive DNA which is there, the number of times it is going to repeat, that is varying. The repeating unit can also vary, as well as the basis in the repeating unit can also vary. Understand this very clearly. Here, ATC is one repeating unit. Clear? I am saying the number of time ATC is going to repeat can vary. Number of time ATC repeats can vary. Okay, first thing. Then the repeating unit, it can also vary. The repeating unit could be like ATCG. This can make one repeating unit. So now what has happened? Repeating unit has changed.
But that can't be in humans, right?
It can be in humans. Why not?
ATCG, that's 4, right?
Also,
You need only 3.
I wrote only 3, matlab?
No, I mean, but like for humans, for coding for the proteins, you need only 3, right?
No, this is not coding, non-coding. This is completely non-coding part, no?
Oh,
You can have a whole strand which is A only.
yeah, got it.
Okay, so what we have to see in the sequence is the sequence is something like this: ATCG, 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 ATCG. So what is getting repeated? That is what you have to check. What is getting repeated? ATCG is getting repeated. Clear? Is that okay?
Yes, sir. Yeah.
And instead of ATCG, can I also write it could be CAG? A, that is getting repeated. Can I change the basis also? Is that okay?
Yes, sir, you can.
Right. So everything within the satellite DNA can change. Is that clear to everyone?
Yes, sir.
So now we have 2 parts of the genomic DNA. We have got the coding DNA, we have got the non-coding DNA, which is basically the heterochromatin part, and we can separate it out into satellite DNA and bulk DNA. Clear?
Correct.
Now, very important thing to notice and understand is
Wait, what are you separating?
separated from the bulk DNA.
How do you separate it?
Cesium chloride density grade centrifugation.
Oh, centrifugation. Okay, fine.
Now, the most important thing which makes the base of this particular DNA fingerprinting is there is variation, a lot of variation in satellite DNA. There is a lot of variation in satellite DNA. Is that okay, guys?
So like variation among the different like people are like in the same.
No, like a particular repeat. Let's say ATC is
Okay.
there. So ATC can be like if I'm talking about a particular human being. So different chromosome can have different repeat depending on how where they have got the chromosome from. For example, I've got my sixth chromosome, one from the mother, one from the father. So father might have a different repeat. I might have a different. Mother might have a different repeat, right?
Yeah.
So now you take 23 chromosomes like that, correct?
Yes, sir.
So 23 chromosomes of a particular type of repeat has come from father. 23 chromosomes from a particular type of repeat has come from mother, giving you a very unique type of fingerprint or very unique kind of profile. Yes or no?
Sir, so can two homologous chromosomes have different codes in the satellite DNA?
Yeah, because that is a non-coding part, no? And even within the coding part also, the alleles are coding for different things, right?
Oh, okay.
It's not necessary. Like, for example, black is dominant on brown. Let's talk about eye color. Okay. So your father's allele for eye color is black eye color. Mother is brown eye color. So they are coding for different thing, right? They're different.
Right. Yeah, correct.
Okay. So there is variation
Yeah,
in the
but
satellite DNA.
within one person is the same, right?
What beta?
Like in one person, that this one's going to be the same, the variation and
Within the same person. No, it can be different.
so the DNA in every cell can be different.
Like DNA in every chromosome, like combination can be different, but it will be same for all the cells.
Huh?
So I understand the variation thing.
See, I'll explain it to you. What I'm trying to say is, first of all, I'm not even explaining the variation part, but first I'll clear what doubt you have. Let's say there are 23 chromosomes, 46 chromosomes, right? Now I am taking example of one. Let's say I'm taking example of chromosome number 5. You've got this from the father and you've got this from the mother, right?
Yeah.
Father का fifth chromosome में the repeat was of let's say ATC and which was getting repeated let's say 50 times. And mother का chromosome में the repeat was of ATG and it was getting repeated 25 times. ठीक है? Is that okay?
Yeah.
Okay. Yeah.
Yes, sir.
Is that okay? Now, what I'm trying to say is, your DNA profile, when I'm trying to make, what will I get? Fifth chromosome, ATC getting repeated 50 times, ATG getting repeated 25 times. So that is your unique barcode, yes or no?
Yeah.
Yeah.
Now, all your cells will have this chromosome.
So they match with my parents also, right?
Half only, right? It will match half. Like your one chromosome will match with father's one chromosome. One chromosome will match with mother's one chromosome. It will not
Okay,
completely, it
yeah.
will never completely match.
Okay. Yeah.
So that's why you are unique. The only place where this completely matches is when you are monozygotic in nature, monozygotic twins. And there is a reason for that. The reason is everybody is formed from the mitosis of a zygote, right? You, me, every human being, first we were zygote, then we underwent mitosis, and then the whole organism was formed, right?
Yeah.
Whatever was the profile of the zygote is the same thing as the profile of my current skin cell or any cell containing the nucleus, right?
Yeah.
So monozygotic twins are what? First the zygote is formed, it divides into two and both of them get placed at different places. So basically they are also formed by the mitosis in the zygote, yes or no?
Yeah.
So unka profile B will also be same, yes or no?
Yeah.
Okay.
So basically, in our chromosomes, half of the repeating thing is from the father, half the repeating thing is from the mother.
Yeah.
Okay.
Hello. Yes. Ma'am, I'm currently in a meeting. I'll call you back. Now, coming back to the variation in satellite, what was I talking or discussing about the satellite में variation क्या होता है? What is this variation? What am I saying? And why is it there in more in the satellite DNA and less in the coding DNA? Let's talk about that. And you're not choosing coding DNA, you're choosing satellite DNA for studying DNA fingerprinting. So there should be some logic behind it, right? So what is the logic? Let's look at the logic. Let's say you have satellite DNA. And this satellite DNA is there in my great grandfather. Okay. Now, here the repeat was ATC, G, ATC, G, and it was getting repeated 100 times. Some random mutation happened, and my grandfather, he received ATCG, and it was getting repeated only 90 times. And to my father, again, there was some mutation, or let's say there was no mutation. He also got it ATCG into 90 times. And when it came to me, I got ATCG. There was mutation, there was some no mutation. I got it multiplied into 80 times. Whatever was the reason. Or I got it 90 times because my father had it 90 times. Let's write it that way. It should happen in the gametes. So I also got it 90 times. So whatever mutation happened, it went on from generation after generation after generation, and it stayed as such. From great grandfather to father to me, the mutation is as such. Okay, we have continued, or I can say that there is a generational pass down of the mutation. Is this okay to everybody?
Yes, sir.
Now, this was satellite DNA. What if there was a coding DNA? And there was some kind of mutation in the somatic cell. Or there was some kind of mutation that has reached up to the germ cell. What kind of mutation? In the coding DNA. There is some kind of mutation in the coding DNA, which has happened either in the somatic cell or it has reached to the germ cell. But that mutation was so dangerous that it killed the cell altogether. Sir, why, sir? Anything that happens to the coding DNA should not be in a way that it is altering the process of transcription and translation. If I am doing anything, any mutation in the coding DNA, there is a high chance that the coding DNA will get affected, and because it is getting affected, it might affect my cell ka survival. Now you tell me, if the cell ka survival is affected, will this mutation reach one generation to the other?
It won't.
No.
It won't, right? So this is one major problem why we do not consider coding DNA, because it is very tough to figure out whether the cell has been continuously, or the DNA has been continuously being passed down from generation after generation. That's why most of the time you will see that there is some or the other kind of relation in between people when it comes to coding. Like we will make some kind of similar protein because that is what is required in order for you and me to survive. We all have the same hexokinase gene. We all have the same allele for or like allele for producing color in the eye or the hair pigment. So what is the reason for that? Because those things are required in order for us to be alive. If there is a mutation there and something happens, the cell dies itself. Is that okay?
Yeah.
I hope till here there is no doubt.
No, sir.
No, sir.
Now look over here. My non-coding DNA or my satellite DNA, I have to talk about that. What are
So
the two
this
types
is
of
just a point mutation that's changing the protein that's being produced.
point mutation? Yeah, it can be point mutation. It could be bigger than a point mutation also.
So will that cell still die?
If the mutation is harmful, then yes, of course, the cell will die, given it happens in the coding DNA, not in the satellite DNA.
What about the case where it doesn't die?
Acha, case where it doesn't die. Let's talk about that. There are 10 people, sorry, 100 people in a population. This you have seen in, there is a line for this in evolution. If the frequency of a particular character is more than 0.1, then it is considered to be a allele, if you remember. There are 100 people. And what do I want? I want one person has has a horn. Aisa, there is a horn coming out of the head. What do you call this?
Horny.
Mutation. What do you call this?
Mutation.
Beta, this is getting recorded. Don't worry. है ना? Second is 10 people start showing this horn. Now it has become a allele or a feature. So when a particular character starts becoming visible in multiple people, then it becomes a allele. Otherwise, it is just a mutation. So when you ask, sir, what happens if there is a mutation in the coding DNA? Beta, if it is letting an organism survive and it is making lots of people with that particular variation or with that particular character, then to it is a proper allele. If it is affecting the reproductive capacity, then it is leading to death of that individual. Is that
So
okay?
that's changing the whole character itself.
Yeah. Let's say because of that horn, some other hormonal changes happen and the person is not able to reproduce. Abhi, they will show the horn, nobody else will show it. Or everybody who had it, they will show it and the next generation will not. Clear?
Got it. Yeah, got it.
Now, there are two types of satellite. There is something called as a microsatellite DNA. And there is something called as a mini satellite DNA. Satellite में single L होता है शायद. Now, microsatellite, the repeating unit is somewhere around 4 to 6 bases or 2 to 6 bases. Repeating unit. This data you can check once, and it is different in different books. And how many times do you see? Number of repeats. Number of repeats is 1 to 6. Not more than that. When I talk about mini satellite beta, which is bigger, mini or micro?
Mini.
So repeating unit here is more than 6 bases. It is made up of more than 6 bases, and number of repeats is also more. Is that okay? Is that clear? This one has more adenine and thymine compared to guanine and cytosine. This one has more guanine, cytosine compared to adenine and thymine. Now comes the part which confuses the people the most, which is short tandem repeat and variable number tandem repeat. What is the meaning of the word tandem? What is the spelling? T-A-N-D-E-M.
Yeah.
Tandem. The meaning of the word tandem is continuous, meaning let's say there is one individual standing over here. This is an orange person standing over here, orange or red, whatever you want to call it. Now, how many people are there? Let's count. 1, 2, 3, 4, 5, 10, 15, 20, 25, 30 and 32. There are how many people, beta?
32.
So right now, if you ask me how many tandem repeats of the red person are there, I will say 32. 32 tandem repeat. Now, let's take one individual in the center over here and convert them into black. Now, if you ask me how many repeats are there, let's say from here to here it is 15. From here to here it is 15. So total number of time orange person has repeated is 30. But the tandem repeat is only 15. 15 tandem repeat here, 15 tandem repeat after that. So the continuous repeating is called tandem repeat. Is that okay?
Yeah.
So now we have understood the meaning of the word tandem. Now there is short tandem repeat. And then there is variable number. VNTR and STR. What
So
is the
this
meaning
still
of both and
under
how is
microsatellite, mini satellite?
uh-huh.
Is this still under mini and
Yeah, it
microsatellite?
is still under microsatellite and mini satellite. Microsatellite shows short tandem repeat and the mini satellite shows variable number of tandem repeat. You'll understand how. So what short tandem repeat is trying to say is that when you have small repeating units which have repeated less number of times, then what you get is called short tandem repeat. But if you have large repeating units which have repeated in a large number of time, then it is called variable number of tandem repeat. So variable number of tandem repeat is seen in mini satellite and short tandem repeat is seen in microsatellite. Is that okay? Is that okay?
So variable number meaning?
Variable number meaning we are not sure how many times it is going to repeat.
Oh, for each tandem.
Yeah.
Okay.
यहाँ तक clear है बेटा? Is this clear till here?
Yeah.
Okay.
Yes.
Now look over here. See what does it say. As stated in preceding section, 99.9% of the base sequence among human is same. It is these differences in the sequence of DNA which make every individual unique. The 0.01%. It is talking about 0.01. Did it cut the? Huh. It is these differences in the sequence of DNA which make every individual unique in their phenotypic appearance. If one aims to find out genetic differences between two individuals or among individuals of a population, sequencing the DNA would be very daunting and expensive procedure. This line is very important. I cannot sequence the whole DNA again and again. Even if I am studying only 3 into 10 to the power 6 base pairs, I cannot sequence the whole thing again and again. Beta 3 into 10 to the power 6 is still a very large number. You cannot sequence the whole thing and read where it is right and where it is wrong. Is that okay?
Yeah.
Sequencing
Yes,
is
sir.
not an option. Then DNA fingerprinting involves identifying differences in some specific regions in the DNA, which is called repetitive DNA. Because in this sequence, a small stretch of DNA is repeated many times. This repeated DNA are separated from the bulk genomic DNA as different peaks during the density grade centrifugation. This we have seen. Bulk DNA forms the major peak. The small peaks are referred to as satellite DNA. Depending on the base composition, length of the segment, and number of repeating DNA, satellite DNA is classified into micro mini satellites. These sequences normally do not code for any protein, but they form a large portion of human genome. These sequences show high degree of polymorphism and form the basis of DNA fingerprinting. Since DNA from every tissue from an individual show the same degree of polymorphism, they become very useful identification tool in forensic application. Further, as polymorphisms are inheritable from parent to children, DNA fingerprinting also becomes the principle for paternity testing. Clear है बेटा? यहाँ तक कोई doubt?
No.
No, sir.
I'm going line by line so that there is no problem whatsoever. Then what is written? As polymorphism in DNA sequence is basic of genetic mapping, both for human genome as well as DNA fingerprinting, it is essential that we understand what DNA polymorphism means in simple terms. Polymorphism arises due to mutation, and different kind of mutations have different effects. New mutation may arise in an individual either in somatic cell or in the germ cells. If a germ cell mutation does not seriously impair individual's ability to have offsprings and who can transmit the mutation, it can spread to other members of the population. Allelic sequence variation has traditionally been described as DNA polymorphism. If more than one variant of a allele occurs at a locus in human population with a frequency greater than 0.01, इसने तो और कम कर दिया बेटा. I wrote 0.1, you can correct it to 0.01. That even if more than one person in a population of 100 shows it, then it is called polymorphism or inheritable mutation. Same logic, same example. Inheritable mutation. And this change
This is the
in
case that
DNA
becomes an allele, right?
हाँ बेटा?
It becomes an allele if it's
Yeah, basically it becomes an allele because it is inheritable.
Okay.
Now, read the next line, which is very important. What does it say? It says probability of such variation to be observed in non-coding DNA sequence would be higher, as mutation in these sequences may not have any immediate effect in the individual's reproductive ability. Because they are not affecting the reproductive ability, that means that they can stay from generation to generation to generation. Is that okay?
Yeah.
Now there is a variety of different type of polymorphism ranging from single nucleotide chain to very large scale changes. Is that okay?
Yes.
Yeah.
Now the technique of DNA fingerprinting was initially developed by Alec Jeffrey. He used satellite DNA as probe that shows very high degree of polymorphism. It was called variable number of tandem repeats. And he also used southern blot and hybridization using radioactive VNTR as probe. Is that clear? Last part means do not look at the technique. Before that, is everything clear?
Yes, sir.
Yes.
Yes.
Now comes the process part, but before that, look over here. The VNTR belongs to a class of satellite DNA refers to as mini satellite. A small DNA sequence is arranged randomly in many copy numbers. The copy number varies from chromosome to chromosome in an individual also. Okay? Within an individual also. It shows various differences. Means ATC could be 20 times in chromosome number 5, but in 7 it could be 10 times. Why? Because you have received it from different parents.
So
Then the
copy
number,
number is same as repeating number.
yeah,
Repeating. Okay.
the number of repeats show high degree of polymorphism. As a result, the size of VNTR varies from 0.1 to 20 kb. 0.1 to 20 kb. What is kb? Kilo basis. So 0.1 into 10 to the power 3 base to 20 into 10 to the power 3 base. Meaning the repeat could be, the VNTR size could be 100 base pair to 20,000 bases. Is that okay?
Yes, sir.
Yes, sir.
All right. So now you have understood the principle. Now let's talk about the technique of DNA fingerprinting. Remember, we cannot do sequencing. Sequencing is not allowed. Sequencing is very costly. Sequencing is a very cumbersome process. We need to do something which can work quickly. Is that pakka clear?
Yeah.
Yes, sir.
Now let's talk about the technique. Let's create a murder scene, beta. Let's create a murder scene. I need 3 people. I'll take random 3 people. Who should I take? I'll take Anant. I'll take Rohan. I'll take Siddharth. Okay. And I've got the police officer as Madan. The thing is, Anant was killed. Where was he killed? He was killed at football field near his home. Okay. The problem was there was no CCTV camera in that football field, and Anant was executed there. The thing is, when we checked the app, Pleo app, we found out that there were 2 people who, 3 people who booked the football field that day: Rohan, Anand, and Sid. Out of the 3 people, 1 person has died, and the other 2 are basically at their home or something like that. So Madan decides to investigate the scene. Madan decides to investigate the scene. When Madan reaches the dead body, first thing Madan says is ki take Anand's blood sample. And send it for DNA profiling. And when Anand's body was being investigated and checked, Madan found out that there is some piece of hair and some piece of skin which is lodged in Anand ka nails. ठीक है, ऐसे nail में यहाँ पे or in the hand, it is stuck. Is that okay? Any problem, beta?
No.
So we will consider this.
So how do they see it like that though?
See it like that means?
Like if I just scratch someone, I won't just like have some skin in my nails, will I?
If you have nails and somebody is trying to attack you and you're trying to defend yourself, then you're going to scratch them, right?
Yeah.
So there will always be a chance of little amount of cell getting stuck in your nails.
Okay.
Or because you were pulling each other, there is a chance that a little bit of hair is being grabbed by the person who is dying. Have you ever been in a situation where somebody is trying to kill you?
Not yet.
Yeah. So you won't know how the body responds when it comes to fight or flight. When it comes to survival, body does different kind of things. So you will try to scratch, you will try to pull, you will try to do whatever you can. So
Yes.
during that struggle, let's say he gets the piece of a hair follicle or he gets a little bit of skin. Okay, whatever. So they get one more DNA sample. DNA sample X. Now, Madan says, being a police officer, he sent this also for DNA profiling. We don't know whose DNA sample is this. Then Madan goes and talks to Rohan and Sid. They both say, yeah, we met Anand that day. We played football also. We had a very good game. And after that, we left. And when we left, Anand was still okay and fine. Then they say, it's okay. Like, we do not know how that happened. And we are really sad and we are really sorry that it happened. But we are not involved. So police basically asks, Bhaiya, the murder did not happen actually at the football field. It happened a little bit nearby towards his home. So did any one of you go and drop him to his home? So Rohan says, no, I had a date. So I had to straight away run from there. So from the football field, I went to HSR. I did not even go towards the house. And from HSR, I went to Kormangla. And Sid is asked the same thing. Sid also says, yeah, I also had a date. So from the field, I went to Marthalli. And from there, we went to Whitefield.
Now I know I am dead.
But I
Okay.
think they killed him together.
Now, the thing is, both of them are saying that we have not been to that area where they were killed. Both of them were in the football field. And after that, Anand went out of the football field. They also went out of the football field. Till here, Madan has figured out. And then somebody has killed them. Somebody has killed Anand on the way to his home. And both of them are saying, no, we are not involved. We directly went to, one went to HSR, one went to Marthalli. Very far away. Hai na? So Madan says, fine, if you guys did not meet him, then there should be no point of your DNA in his nail. So what we do is we will take DNA sample from Rohan and Sid also.
So, but how are they so sure that it was gonna be from the crime scene and not while playing football?
Why would you pull somebody's bunch of hair, or why would be their skin tissue in
So
your
you
hand like that?
can't play
It's a
football.
game is game.
You
Game is
can't play
game.
football
What are you
with
playing?
us.
Football or something else?
Yeah. Sometimes you just scratch other person badly.
Calm down, champ. Calm down. No, we asked, no, was there any fight in between the game? Rohan and Sid both said, no. It was a very like a long distance game. We were just passing the ball around and we went home. Okay. So DNA sample was taken. Let's label it this as Rohan and this as Sid and this one as Anand. Now, how do you profile the DNA? What are the steps for profiling the DNA? Let's look at that. First thing that you do is you isolate the DNA. Have you guys studied in biotech how to isolate DNA?
Yeah, gel electrophoresis.
Arey, that is not gel electrophoresis. How is it?
Ari.
What is RE?
Oh, you mean before gel electrophoresis?
Restriction endonuclease.
You don't do a restriction endonuclease. Then how do you take it out from somebody's nucleus?
So
Oh, you
Elution. What is
use
it?
pooling or
protease
something.
and
No?
all.
Spooling. Very good. What do you use? Spooling. First, you digest the outside membrane by lipase, protease, and all of that. And finally, you give chilled ethanol to it and then you spool the DNA out. Is that okay?
Yeah.
Yes.
Now, if the DNA content is less, if it is less, then you use PCR. Is that okay? Is it
Yeah.
fine?
Yes, sir.
Once you have got enough content, then you use any restriction enzyme to break it down. Use RE to break down DNA into small parts. Is that okay? Once you have broken it down into smaller part, then use your gel electrophoresis. I hope everything till here is clear. Okay. And when you are separating the fragment in the buffer, gel electrophoresis is buffer में, use sodium hydroxide so that DNA becomes single stranded. I'll tell you why we are making it single stranded later. Is that okay? Clear? Any doubt
Yes.
till here?
No, sir.
We are doing this for everything. We are doing it for the Anant ka DNA, we are doing it for Rohan ka DNA, Sid ka DNA, and we are doing it for the DNA sample X which has been found in the crime scene. Is that clear?
Yeah.
We have done it for all the 1, 2, 3, 4 samples. Then the next thing is from the gel electrophoresis, what you do, जो agarose plate होता है, the agarose plate which is there, on top of that agarose plate, what do you place? You place nitrocellulose paper. And then you press it better. जैसे if this is your gel electrophoresis plate and this is your nitrocellulose paper, you put some paper weight on top of it so that what happens, that the DNA, it basically gets stuck or is transferred from the gel electrophoresis plate, the agarose plate, to the nitrocellulose paper. This process is called southern blotting. So if somebody asks you what is southern blotting, beta, what would you say? It is the, what is it?
Transfer of the genetic material from the
Transfer of the DNA. Remember, transfer of DNA, not the genetic material, transfer of DNA from agarose plate to nitrocellulose paper. Is that okay?
Yeah.
If you do the same thing with protein or with RNA, if you do the same thing with RNA to nitrocellulose paper or protein to nitrocellulose paper, like you press it and you transfer it. For RNA, it is called northern blotting, and for protein, it is called western blotting. Nobody's going to ask you in detail. Is that clear?
Yeah.
Yes, sir.
Yes, sir.
Now, what you have to do is, you have to take the DNA from sample X. Okay. This is your DNA sample X, the one which was found in the crime scene. Okay. And you have to find out the satellite DNA sequence of this DNA. Whatever is the repeating unit, we just have to figure that out. See, let's say in the satellite DNA, for this particular case, it was ATCG, ATCGA. This is the repeating unit. Is that clear?
Yes, sir.
Yeah.
Now we have already made the during the gel electrophoresis, we have made the DNA single stranded, and that is how we have kept it on the nitrocellulose paper also. And now we have found out that the DNA sample in the X, the satellite DNA is ATCGA. All you have to do is create an artificial probe, artificial radioactive probe. Sir, what is an artificial radioactive probe? Here, you will create a complementary DNA to the satellite DNA with a radioactive phosphorus or radioactive sulfur attached to it. So what does it look like? it will look like this. A ka complementary is TAGC, TAGC with something radioactive labeled attached to it. This is what you have to create. Is that okay?
Yes, sir.
Yes, sir.
Then you take all the 4 DNAs which you have taken. Okay. You take the Anand ka DNA, the crime scene ka DNA, Rohan ka DNA, and Sid ka DNA. You keep all the 4 in 4 different packets.
So, but why is Anand's DNA even required?
We are not sure na ki kya pata wo uska khud ka hi DNA ho, one which is there in his nails.
Oh, okay.
Like
Yeah,
what if he scratched himself?
So we take the artificial radioactive probe of the one that's found in the fingernails, no?
Yeah, on the fingernails. That is the one that we are doubting. So we take it. We take the 4 packets of the 4 different DNA that we have. Now, in the 4 different package, what we do? We have added the nitrocellulose paper which is there, containing DNA of Anant, DNA of sample X, and DNA of Rohan and Sid. After that, we have added this is a Ziploc packet. After that, we have added the probe. What have we done, beta? We have added the probe. Is that okay?
Yes, sir.
After adding the probe, we have added it in the packet, and we have to shake that packet so that in case if there is any chance of attaching to the single stranded DNA, it will attach. You know that it will only attach to the single stranded DNA if it is completely complementary. So once we do that, we remove all the content from the Ziploc packet, and we have the nitrocellulose paper left with us, which we have to watch under X-ray film. Now this is the moment of truth. What have you done? In the packet, you have taken your nitrocellulose paper of DNA containing samples from all 4 places. You have added the radioactive probe. You have shook it well. After that, you have removed the excess content, and now you are watching it under X-ray film. One place from where the bright color bands will be seen is the sample X, because we have used it as our source of probe creation. Right? इसी को तो use किया था ना for making complementary probe. Yes or no?
Yes,
Yeah.
sir.
And now we see that in Anand's case, there is no visibility of any radioactive film. In case of Sid also, there was no visibility, but in case of Rohan, there was again visibility of colorful bands. So we go and ask Rohan, beta, you said ki after your game, you directly went to HSR and from there you went to Koramangala. How come there is your skin sample in Anand's mouth and his your क्या बोलते हैं fingernail? Did he try to bite you and try to scratch you up when you are trying to kill him? So once this is proven that the crime scene DNA and the suspect DNA matches, then we take that person into investigation room, and after that we do the things from there. Like Madan will beat him up and figure out how the crime was committed. Is that clear?
Yeah.
Yes, sir.
Is that okay?
Yeah.
Yes.
Now look over here. What is the process which is given? Isolation of DNA, digestion of DNA by restriction endonuclease, separation of DNA fragment by electrophoresis, transferring the separated DNA into synthetic membranes such as nitrocellulose paper, hybridization by using VNTR probe, detection of hybridization during autoradiography. After hybridization with VNTR probe, the radiogram gives many bands of different sizes. This band gives a characteristic pattern for an individual. It differs from an individual to individual in population, except in case of monozygotic twin. That's why the color came only for Rohan's DNA. Consequently, DNA from a single cell is enough to perform DNA fingerprinting analysis and in application to forensic science. It has much wider applications, such as in determining population and genetic diversity, and currently many different probes are being used to generate DNA fingerprinting. Look over here. DNA for crime scene is obtained. Let's use this and this. है ना? Crime scene में you have got the DNA, and this is what the bands look like. This is chromosome number 16. This is chromosome number 2. This is chromosome number 7. Crime scene में the DNA that you have got This is what it looks like on the film. This is the film. These are DNA individuals or suspects from individual A and B. All you have to do is match. Check where it is matching. Is B matching or is A matching?
So B.
See, this is matching with this. 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 And this is matching with this. Is that okay?
Yes, sir.
Is that clear?
Yeah.
Is DNA fingerprinting clear now?
Yes.
Yes, sir.
Any doubt, anyone?
No, sir.
All right. Now next is your Human Genome Project. Want a break of 5 minutes?
Yeah.
Yeah.
All right. Human Genome Project is comparatively a easier topic. When you look at DNA fingerprinting and Human Genome Project, it is comparatively a easier topic. The only thing is there is lots of data that we have to study. You know? And it was a mega project. It has lots of things to take in consideration. There were a lot of people who were involved. There were so many goals and there were so many results and all of that, salient features and everything like that. The only thing where you get confused is the methodology. If I am right, that is the only point of concern. Yes or no? Hello, hello. Guys?
So we didn't do it yet. Are we supposed to know?
Huh? What did you
We
say?
actually don't know. So can you hear now?
Yeah. Tell, what did you say?
Are we supposed to know it?
Human Genome Project.
Yeah, I mean, we don't know it.
Yeah, it's not been done in class yet.
She took like one online class, extra class for it, but no one joined.
So no one joined.
Then we'll do it properly. Pura starting se karenge. Bhai. First of all, let's understand what is the meaning of the word genome. Any idea what is the meaning of the word genome? Genome means haploid set of DNA. If the diploid set is 46 chromosome, the haploid set is 23. If the diploid set is 6.6 into 10 to the power 9 base pair, the haploid set is 3.3 into 10 to the power 9 base pairs. What we need to understand is the DNA sequence is what makes a person. How? DNA sequence codes for protein, and it is the protein that is basically showing, or it is a protein which is basically present, or I can say protein is the expression of the characters. So whatever form of expression you see, it is because of the DNA sequence. So if I look at me, if I look at another human being, what I'll notice is that people are very different. So this basically pushed the human brain into thinking ki, bhai, what is that separates us out? What if we figure out the complete thing? What is that makes our complete sequence? We need to figure that out. And by the time this started, basically back in 1990, you had all the technology. You had all the technology of biotech. Tools of biotech were available to you. Biotech tools available. So using those biotech tools, we decided that we will analyze all the sequences. When did we start it, beta? We started it in year 1990. And it ended in 2003. So how long of a project it was? 13-year long. How many years, beta?
13.
13 years. Okay. What are we sequencing? Acha, and what is sequencing? Beta, sequencing is basically finding out the arrangement of base pairs. Finding out arrangement of base pairs.
So even in fingerprinting, you need sequencing, right?
No, we don't do sequencing. We just get it. We just get the sequence of the satellite DNA to make the radioactive probe. We do not do sequencing at all. It's a very cumbersome process, a very long process.
No, but then sequencing for that particular sample DNA.
For the satellite DNA.
Huh.
Even in the sample DNA, just the satellite part, the repeating part,
Oh,
that's it.
okay.
And most of the time, we have already created repeats with us. We just try it out. हमारे पास already known repeats are there, so we can try it out.
Okay.
Then you have how many bases to sequence? 3.3 into 10 to the power 9 base pairs. This is how many base pairs you have to sequence. Is that okay?
So shouldn't
How
be
long was it?
6.6?
No, we are doing the genome sequencing. The genome is
Okay.
half.
Okay, fine.
And 1990 to 2003 is the whole project, but chromosome 1 was so big that the sequence happened till 2006. Clear? "22 autosomes, 1 X chromosome and 1 Y chromosome." DNA was not obtained from a single individual, beta. It was obtained from different individuals, and the identity of those people have already always been kept private. Like nobody knows who those people are, but mostly they were from European origin only. Now, I have to tell you how big of a project it was. How big of a project, beta? If one base का sequencing costed $3, $3. Array, write properly, no? The money would come out to be somewhere around 9 billion US dollars. Sir, 9 billion is not that much, no? Like our Musk and all, they're at 143 billion and stuff. But back in 1990, it was a big deal. है ना? अभी तो economies have opened and everything is fine. But back then, it was a big deal. Sir, is there any other way other than money to compare and tell us how big of a project it was? If I take a book with 1,000 pages, ठीक है, and every page has 1,000 letters, then I will need 3,300 such books to store all the data. That is how big of a project it was. So did we actually do it in books and stuff? No, beta, we did not do it in books. We had a program for it. We put it in computer. Sir, how big of a project, sir? We are not understanding it by the money part. We are not understanding it by book part. But it was so big that a whole branch of science now exists because of it. A branch of biology to deal with this, to sequence. Branch of bio to deal with. Branch of bio to deal with data storage and retrieval. What is this branch called? Bioinformatics. Sir, so what, sir? A new branch has been formed and so many books have been used or so much money have been used. So what, sir? We are still not getting how big of a project it was. Okay. Then I'll tell you the people who are involved. So many people are involved, right? So first, agencies involved. You have got your US Department of Energy. US Department of Energy. Then you have got National Institute of Health, the NIH, again from USA. Then you have got Wellcome Trust of United Kingdom. Then you have got involvements of country like Japan, France, Germany, and China. So you've got these many countries who were involved. So a project that took somewhere around 13 years and 3 extra years for chromosome 1, a project that involves sequencing 3.3 into 10 to the power 9 base pairs, a project that involves sequencing 22 autosomes, 1 X chromosome, 1 Y chromosome, a project that involves somewhere around expenditure of $9 billion, more than that, somewhere around 10, 9 is what is there. A project that involves use of 3,300 books. If hypothetically we are storing the data in the book, a project that involves formation of a new branch to deal with it, a project that involves US Department of Energy, National Institute of Health, Wellcome Trust of United Kingdom, Japan, France, Germany, and UK. So all this together makes up the Human Genome Project. Is that clear?
Yes, sir.
Now, is there any other organism whose genome has already been sequenced? Answer is yes. We have already got genome sequence of Saccharomyces. You guys will tell me what is Saccharomyces? What kind of organism is it?
Yeast.
What is it, Saccharomyces?
Yeast.
Yeast, which
Fungi.
is a type of fungi. And we have already got it for E. coli. We have sequence of Drosophila, the fruit fly, the one which was used in linkage experiment. We have it for Caenorhabditis elegans, C. elegans, which is a nematode roundworm.
So
Then
should
we
we
have,
know all the agencies involved?
yeah, we have got it for rice, and we have also got it for Arabidopsis. Spelling एक बार check कर लेंगे, ठीक है? So this is the insect, this is a roundworm, these two are plants. So these are the organisms whose sequence has already been done, and along with that, human can also be included. Is that clear?
Yes, sir.
Now, what were the goals? What were the goals? Let's talk about the goals. Goals के लिए, should I write it down or should I just read it out? I'll read it out. This is the salient features. Goal is not there, yeah? Goals are there. What were the goals? Goals were to identify all approximately 20,000 to 25,000 in the human DNA. They were thinking that there are somewhere around 20,000 to 25,000 DNA. Determine the sequence of 3 billion chemical base pairs that make up the human genome. Okay? 10 to 10 to the power 9. Store this information in some database. Improve the tool for data analysis. So they didn't just want to store the data, they wanted to analyze it and to use it. Transfer the related technology to other sectors such as industries so that it can be used. Arey bhaiya, you are sequencing human DNA, at least use it for making some medicine, some industrial purposes and all of that. On top of that, they wanted to address the LC. What is LC? Ethical, legal, and social issues. When you start something, there will always be a question that, bhai, are you doing it with good intent or bad intent? What if you figure out some information and you use it to create a biological or a chemical weapon? What if we find out the sequence of certain people and we get to know a particular community? Let's say the Asian community is more prone to getting a particular, more prone to a chemical or more prone to a virus or a bacteria. And what if we start a biological warfare? So there is always going to be some ethical, legal, and social issues. So we have to address that as well. Is that clear?
Yeah.
है ना? What else do we know? In the preceding sections, you have learned that it is the sequence of bases in the DNA that determine the genetic information of a given organism. In other words, genetic makeup of an organism lies in the DNA sequence. If two individuals differ, their DNA sequence should also be different, at least at some places. This assumption led to the quest of finding out the complete sequence of human genome. With the establishment of genetic engineering techniques where it was possible to isolate, clone any piece of DNA, and availability of simple and fast techniques of determining DNA sequences, a very ambitious project of human genome was launched in year 1990. It was called the mega project. This word is important. Remember it. Not micro, not nano, not macro, anything like that. It is mega project. You can imagine the magnitude by the following. Human genome is said to have approximately 3 into 10 to the power 9 base pair. If every base pair charged $3, the project would cost somewhere around 9 billion US. If the sequence to be stored in a book containing 1,000 letters per page and 1,000 pages per book, 3,300 such books would be required. And the required to store the information from single genome. An enormous amount of data to be expected to be generated, and use of high-speed computational device for data storage. HGP was closely associated with a rapid development of new area of biology called bioinformatics. Then, Department of Human Genome Project was a 13-year-old project coordinated by US Department of Energy, National Institute of Health, Wellcome Trust of United Kingdom, and contribution came from Japan, France, Germany, and of course China and others. The project was completed in 2003. Knowledge about the effects of DNA variation among individuals can lead to revolutionary new ways to diagnose, treat, and someday prevent thousands of disorders that affect human beings. Besides providing clues to understanding human biology, learning about non-human organisms, DNA sequence can lead to understanding of their natural capabilities that can be applied to solving challenges in healthcare, agriculture, and energy production. Beta, ye yaad rakhna. What they do is they'll give a very random statement that Human Genome Project can be used to solve these things. Hai na? So you have to remember this.
Yes, sir.
Many non-human models such as bacteria, yeast, Caenorhabditis elegans, Drosophila, and plants like rice and Arabidopsis have also been sequenced. So you have to remember their name. कौन कौन है बेटा? E. coli, Saccharomyces, the nematode, Drosophila, plants like rice and Arabidopsis. Clear? Clear?
Yeah.
Yes, sir.
So goals we know. Now let's talk about the methodologies. Whosoever will get confused here only. There is no other point of worrying about HGP. It is majorly ki read it 5 times, 10 times, and you are sorted. Now, there are two methodologies. Methodology number 1 is called express sequence tagging. In short, we call it EST. Any problem, beta?
No, sir.
Express sequence tagging. Let's understand how it works and what is the logic behind it. Okay. One approach is this. Second approach also whole genome sequencing we will see. But first, let's understand what is the scene here. You know, we are talking about eukaryotic cell, yes or no? Yes or no? Are a human cell is eukaryotic, yes or no?
Yes, sir.
Yes, sir.
Sir.
So whatever DNA is there in the eukaryotic cell, it is going to be double-stranded DNA. Yes or no?
Yes, sir.
And the DNA first converts into a type of RNA which is called hnRNA. Yes or no?
Yes, sir.
And the hnRNA then gets what? It's
Nice.
shutting done and post-transcriptional modification, and then it turns into the mRNA which is brought into the cytoplasm. Yes or no?
Yes, sir.
Yes, sir.
Now, beta, this mRNA is the one which is actually going to get converted into protein, right?
Yes.
Yeah.
Does this mRNA contain any exon? Contain any like random segments like intron? Does it contain any intron?
No.
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 Does it contain any extra random sequence which we don't want?
No.
No.
It is purely mRNA, right? It is purely mRNA which is free of all the problems. You can take it, you can convert into protein. What if What if instead of going into the nucleus, taking out a whole segment of DNA, doing the sequencing, instead of doing all that, what if I don't even get into the nucleus? What if I come to the cytoplasm? Where do I come, beta? Cytoplasm. And in the cytoplasm, what if I take all the mRNA and use something called as reverse transcription? And after using reverse transcription, what if I make this mRNA into a double-stranded DNA? Now, this double-stranded DNA that I have created, when I make it undergo transcription, will it make mRNA directly or will it make hnRNA?
mRNA.
It will make mRNA directly. This kind of DNA which makes mRNA directly for you is called cDNA. What is it called, beta? It is called cDNA or complementary DNA. Now, when you do this, you are basically looking at all the coding DNA, all the DNA which is making RNA. You are not looking at what? You're not looking at what? You're not looking at the non-coding part at all. Is that clear?
Yes, sir.
So if you sequence cDNA, basically you are sequencing the express DNA. Which is getting expressed. So can I simply not call it express sequence tagging? Once you have got the cDNA and you know what function it is doing, you can do the annotation ki bhai, this cDNA is doing this work. This is called sequence annotation. What is annotation, beta? When you annotate a PDF, you take a line and you write what is the function of it. So you take the DNA sequence and you write this is the function. This one makes insulin. This one makes growth hormone. This one makes testosterone. This one makes tryptophan. This one. That is called annotation. And this whole process is called express sequence tagging. Is that okay?
Yes, sir.
So this was one approach towards your Human Genome Project, that do not sequence the complete thing, just sequence whatever is required. Is that clear?
Yes, sir.
Then look at this. Are you guys writing all of this?
Yeah.
Good, beta. And for those who are not writing, I'll not send the notes.
So didn't you already give the notes?
It is different. It's far more better. This is far more crazier. Then it's not like that. Of course, there will be few points here and there when you teach it is different because the examples I use and all of that. But yeah, the whole genome sequencing part, let's talk about. Here, we do not care whether you make the RNA, whether you make the protein or not. We are going to sequence everything. Both coding and non-coding regions are sequenced. Both coding and non-coding regions are sequenced. Is that okay? Is that clear?
Yeah.
Yes, sir.
But why would they do
How-
this if it's better to just sequence the mRNA?
See, the problem is we want to study the whole sequence in order to understand the, not just the coding part. We need to know the origin of DNA. We need to understand the chromosomal structure, how one part is affecting the other. For example, satellite DNA, how is it affecting the fingerprinting? Jab you will not even study, then how would you even know?
Correct.
Okay. So we want the complete thing. All right, so we want the complete thing. So, sir, how do we do it? First, again, let's take a cell. This is our dear cell and this is our cell ka nucleus. And inside this, what do we have? We have the DNA. because it is double stranded. So we will duplicate and make it like this. You have to, first step you have to do is isolate the DNA. We have already seen how to isolate the DNA. First, you add lipase, protease, RNAs, and all of that. And then you add chilled ethanol, and the DNA is isolated. Clear? Clear?
Yes, sir.
After that, we let the restriction endonucleases act on it. Restriction endonuclease act on it. Just a second, guys. Somebody's knocking on the door. Okay, coming back to, so what are we doing? We are basically taking the DNA out, and isolate the DNA and apply restriction endonuclease on top of it. So let's say this is our DNA. ATCG, ATCG, TATA, GCGC, TCAT, CG, ATT, TC, GATG, AT. Random sequence I've taken. Okay, I've not taken some something which is like actually there. I've taken a hypothetical. Now, earlier what we used to do is we used to teach the fact key. We are using one restriction enzyme, then we are separating the fragment, then we are using another restriction enzyme and separating the fragment. But the problem is NCERT talks about something called as, I'll show it to you over here. The sequence were arranged. The sequence were arranged based on some overlapping regions present in them. This is where I felt like it is talking about something which you need to understand in a different way. We cannot go by just 2, 3, 4 different restriction enzymes. You need to understand and grasp this concept really, really, really well. Let's say this is the sequence over here, and I have put random restriction enzymes. One is cutting over here. Okay, one is making a cut over here. Acha, after the first one's cutting, what do we get? We get the fragment which looks like this. थोड़ा दूर काटते हैं इसको. We'll cut it somewhere over here. The other one is cutting somewhere over here. Then one is cutting over here. And one is cutting over here. And there are different restriction enzymes. You have let them go. So they have cut at different points. Let's name it A, B, C, and D. And after cutting, these are the fragments that have been observed. What are the fragments? You have got one fragment which looks like this. Then after this one's cutting, we have got the fragment which looks like this. I'll change the color so that you understand clearly. Sorry. Then you have got one which is cutting over here. Where was it? CGATTD, right? So one sequence will look like this. Just give me a time, okay? It's for your understanding only. And then the last one. You know, so the green color ones are because of restriction enzyme A. This is because of restriction enzyme B. This is for C and this is for D. Is that okay?
Yes, sir.
Okay. Now, you do not know what the actual sequence is. Why, sir? Because beta, we have taken out the fragments. We do not know. In the reality, in the real picture, we do not know whether this goes with this or the other thing goes with the other thing. Where did it go? Huh. Is that okay? We do not know which fragment is going with which fragment. Is that clear? Is that clear, guys?
Yes, sir.
Yeah.
Okay. But what we are sure about is because we have cut the same fragment, there will always be some overlapping regions. For example, look at this fragment. Isn't it matching with this one up till here? Let's take out the fragments and arrange them properly. This is the first fragment, right? So if we have some kind of software, then we can look at it this way. If this one is getting really similar to this, which is again really similar to this, which is again really similar to this. And if we look at the other fragments also, what we will notice? The other fragments are also, where is it, Tata ji? It is really similar like this. This is really similar to this part. This is literally or really similar to, where is CGA triple T? CGA triple T over here. And this part. So what we have done is based on the overlapping fragments, because they were overlapping in nature, we played the jigsaw puzzle and we were able to make this particular sequence, yes or no? Tell me that.
Yes.
But the only thing is ki beta yahan pe there was somewhere around only 20-25 base pairs, no? So I was able to do it by hand. This one will go with this one will go with this one, and this one will go with this one. In reality, how many base pairs are there? 3 into 10 to the power 9, right?
9.
If there is 3 into 10 to the power 9 base pairs, will a human being be able to do it?
No.
No.
So based on this overlapping fragments and this possibility, there was a guy named Frederick Sanger. Frederick Sanger. He was also responsible for figuring out what is the code of amino acids from the DNA. We'll study about that. So he was the guy. He created a software, and in that software, when you added the overlapping sequence, they were able to figure out who goes with who. So basically, they arranged the whole sequence. Is that clear?
So your
Yes, sir.
voice broke. Could you just explain the overlapping part again?
Which part?
The
Where did I?
overlapping.
What was the last line that you heard?
Like overlapping. I didn't understand why they're overlapping.
They're overlapping because beta, it is the same fragment which has been cut by different restriction enzymes. Like over here, did you understand the explanation part where I made the whole diagram?
No, your voice was breaking.
Okay. So what we can do is we can go like this.
So did they cut the whole human genome?
First, they broke it down to smaller parts, they copied it, and then they cut those.
Oh,
So they did it for smaller parts, then they arranged them all together and they got the whole genome.
okay.
Look over here, wait. This is the sequence. This is your restriction enzyme A cutting over there. Can you see?
Yes, sir.
A, B, C, and D. Is that visible?
Yes, sir.
Now, let's we have removed the क्या बोलते हैं DNA sequence now that we have to build, and we are just looking at the fragments. Let's take the fragment one by one. Now we are going to arrange the fragments one by one. This ATCGTACG was basically formed when the restriction enzyme A cut it, right?
Yeah.
So now when we try to overlap it, the next fragment which was cut by B is overlapping up to a certain part, yes or no?
Yeah, yeah.
The blue is overlapping on the green, yes or no?
Yes, sir.
And then the red is also overlapping little bit more.
Yeah.
And finally the yellow is also overlapping.
Yeah.
Then we look at the next part. A part of yellow and red is being overlapped by green.
Yeah.
And then a part of this thing. What is it called? Red, no? Next is red. A little bit of yellow is overlapping the green, then red is overlapping it, and then blue is overlapping it. And
Yeah.
finally, when we bring all the pieces together, it looks like our original DNA sequence that we were talking about.
Yes, sir.
So this is what I was saying. In reality, what happened was they took the DNA, the whole DNA, then they broke it down into multiple fragments. They took those fragments and they made multiple copies of them, and they started doing this. They started cutting them by restriction enzymes, started separating them out, putting it for Sanger sequencing. What is Sanger sequencing? It was a mechanism developed by Frederick Sanger, where he used this overlapping fragments to create the whole piece. It was basically like the jigsaw puzzles. So you had the puzzles and the part was overlapping, so it will go like this. It will fit. So basically on the basis of whether it overlapped or not, whether it fit properly or not, you got the whole sequence. Is that okay now?
Yes, sir. Got it.
Now look over here. Look at the methodology. What does it say? It says the method involves two major approaches. One approach is on identifying all the genes that are expressed as RNA. This is called express sequence tagging. The other was the blind approach of sequencing the whole set of genome. The whole set of genome contain all the coding and non-coding sequence and later assigning different region with sequence and function. Pehle they are saying that we will sequence everything, and on the basis of what we have sequenced, we are going to annotate it that this part is coding and this part is non-coding. Is that okay?
Yeah.
Then you have got for sequencing the total DNA from a cell, it is first isolated and converted into random fragment of relatively smaller sizes and then cloned in by suitable hosts using specialized vectors. If you have studied a little bit of biotech, then you would know that there are two ways of cloning any DNA fragment. Either you can put it in PCR or you can use vectors. What are vectors? These are basically plasmid-like of things, where you put your choice of DNA and as much time the plasmid copies, that many times your DNA copies. Is that okay?
Yes, sir.
And the vectors that were used, they were called BAC and YAC. Just straight up remember their names. Reason for using them was DNA was big, fragmented. DNA was very big. So this fragmented DNA, which was basically very big, this fragmented DNA needs to be copied. So you cannot use your normal plasmid. So you have used back and EAC. Is that clear?
Yeah.
And then fragments were sequenced using automated DNA sequencers that worked on the principle of method developed by Frederick Sanger. Sanger is also credited for developing method of determination of amino acid in the sequence of protein. What amino acids are there in the protein? That was also developed by Mr. Frederick Sanger. Sequences were then arranged based on some overlapping regions present in them. जो अभी हमने देखा ना, whatever we saw right now, that is what happened, but it happened in the computer, in a software, in a technique developed by Sanger. This required generation of overlapping fragments. Alignment of these sequence was humanly not possible. That's why you had computer-based program to do this. The sequence was subsequently annotated, assigned to each chromosome, and the chromosome 1 sequence was completed only in May 2006. If I want a very good statement-based question, I'll change it into September 2006. So you will get confused if you have not read it properly, that whether it was May 2006 or September 2006. How do you remember this? Very simple. NEET kab hota hai beta? NEET happens in which month?
May.
Chromosome sequencing done in which month?
May.
As simple as that. Abhi wahan pe NEET on the day of the exam, if it comes by chance ki bhaiya this was done on which month, you just remember that sequencing ended on the same month when we write the NEET exam. We write the NEET exam in May, so we have to remember. And the 24 chromosome that they studied was 22 autosomes, 1 X and 1 Y chromosome. Then the next task was how to make
the overlapping will happen because of you're taking fragments from different
restriction enzymes. Like the same fragment cut by different restriction enzymes.
Oh, same fragment by different restriction. Okay.
Yeah. Then look at the salient features of human genome. It contains 3,164.7 million base pairs, which basically translates to 3 billion base pairs. Average size of gene is 3,000 bases, but it can go up to largest known gene with 2.4 million bases. Total number of gene is estimated at 30,000, which is much lower than the previous count of 80,000 to 1,40,000. And almost 99.9% nucleotide bases are exactly same in all humans. The only difference is 3 into 10 to the power 6, which we study in DNA fingerprinting. Then function of over 50% of discovered gene is unknown. We don't even know what their function is. Less than 2% of the genome actually codes for protein. Less than 2% means what is 2%? 2 by 100 into 3 into 10 to the power 9, which comes out to be 6 into 10 to the power 7. So less than this. Okay. Then repeated sequence make up very large portion of human genome. Acha, here we can make one line key. All the repeated sequence is not satellite. All satellite is repeated, but all repeated is not satellite. Repeated sequences are stretch of DNA sequence that are repeated many times, sometimes 100 to 1,000. They have no direct coding function, but they work on chromosomal structure, dynamics, and evolution. It is said that these were basically coding for something. Now they are not useful anymore. And it is also said that they provide stability. Then chromosome 1 has most genes and Y has the fewest. Scientists have found out 1.4 million locations where there is single base, single nucleotide change. Okay, this information promises to revolutionize the process of finding chromosomal location for disease-associated sequences and tracing human history. Best example of understanding this is sickle cell anemia. In sickle cell anemia, what happens? There is only one point mutation, right? Remember or not?
Yeah.
So similarly, we have found out 1.4 million locations where there is only one single nucleotide ka change. So deceased person and non-deceased person ke beech mein whatever differences, we can figure that out. Yes or no?
Yes, sir.
Yes, sir.
Okay. And there are more other, there are other ways of using it also, but apne syllabus ke liye only this much is fine. Is that clear?
Yes, sir.
Now there is a lot more talking about where we can use this human genome sequence. We can use it for biological research. We can use it for treating cancer and all those other things. So notes for this topic. and the DPP for this topic will be shared to you very quickly as soon as possible. Do that, ask your doubts. I'll stop recording now.